Okay, the last part of protein synthesis is when mistakes happen, and so we can have mutations. So what are mutations? Mutations are changes in the genetic material. And then remember, the genetic material contains instructions, the order of the bases to make polypeptides, and so therefore it can change your proteins that you make in your cells and change the traits of an organism. Mutations can cover large portions of DNA, or they can be a point mutation, meaning that it is just one particular point or at one particular base is changed. There are two broad categories of point mutations called base pair substitutions and insertions or deletions. So what causes a mutation can be caused, as it says here, by a number of things. Errors in replication. So when DNA is replicated, the A's are supposed to go with T's, C's are supposed to go with G's, and so on. So errors can happen there. Recombination, so like crossing over, or when they repair their DNA, and we'll talk when DNA replication occurs, if there is an error, sometimes it can be caught by specific enzymes and repaired, but if it's not repaired correctly. And then things called mutagens. These are things that are known to change your DNA. So UV radiation, certain chemicals mutate your DNA. So those are known things. So let's look at this picture here. We have wild type hemoglobin DNA. So this is DNA to code for making the protein hemoglobin, which remember is in your red blood cells that helps us to deliver oxygen to our cells. So this is the template strand. It's your template DNA. So this is the 3' end, this is the 5' end, which means that the mRNA will be built from left to right so that the mRNA is grown from the 5' to 3' direction. So therefore, here's your mRNA. So what we're looking at is just the codon, the series of 3 bases that are important in the mutation that occurs. And so normal mRNA, this is supposed to be the 5' and this is the 3' end. It's built in the 5 to 3' direction. And so the bases match up, and so the G goes with it, and T and A, and so on and so forth. So we make the mRNA. So from here to here is what process? DNA to mRNA, transcription. From here to here, now we go from the mRNA, and we're going to hook together amino acids. So these are supposed to be amino acids, and this is glutamine, GLU. And so this is your chain, and this is much longer. It's a ... All right, and so the mRNA codon GAA stands for glutamine, GLU. So that's the amino acid that's supposed to be in that spot. So mRNA to amino acids, this is translation. Only thing this is not showing you is the RNA processing where the introns are cut out. So then let's look at what happens with the mutant hemoglobin. This is in sickle cell disease. And so here's a person who, their template DNA, notice here it's supposed to be CTT, now it's CAT, so the T has mutated to an A, and that changes the mRNA's codon. Instead of GAA, it's going to be GUA, and then if you look at your chart, GUA, if we go back to the chart, because remember we used the chart with mRNA codons, so we go back here, A is valine. All right, so now that's why it says here in this spot it's VAL. We have a different amino acid. And if that amino acid is important in like the 3-dimensional structure of the molecule, remember when you get a polypeptide chain and it goes like this and the folds and the R groups interact and so on. So if that amino acid is important in determining the shape and function, therefore, of the polypeptide or the protein, then you're not going to have the right function. And so that's what happens in this hemoglobin. That valine is an important amino acid in determining the shape of the hemoglobin molecule. So remember, it's sickle-shaped in people, all right, makes the red blood cell sickle shaped, and so therefore interferes with the job of the hemoglobin, which is to carry the oxygen to the body cells. So let's look at different types of mutations. We said that there's two categories, base pair substitutions and insertions and deletions. So let's look at base pair substitution. So where you substitute one base pair for another, hence the name. So one base pair gets substituted for another. Sometimes these are silent because of the redundancy of the genetic code. So what does that mean? If they're silent, it means that just because you have a mutation doesn't necessarily mean that we change the amino acid. So for instance, the redundancy of the genetic code, if we go back to the chart over here, this is what I was talking about with the third base here, that CCU, if it got mutated to CCA, it doesn't matter because it still stands for the same amino acid. And so therefore proline here, there's four different codons that could code for it. And so that last base could be mutated and it doesn't matter. And even though a mutation occurs, it doesn't matter. And so therefore they call those silent mutations because you don't get a side effect from it because it codes for the same amino acid. All right, so that's the redundancy of the genetic code. So maybe we should write that down here. So example, proline. We used that example before, but proline is CC. The first two are CC, and then you can substitute any other base in here, and it's still proline. So that's the redundancy. Other times they are silent, they meaning the mutations, because the mutation codes for an amino acid or protein that is not essential to the protein's function. So if that amino acid has nothing to do with determining the structure and function of the protein, you may not see a side effect. It still may have enough amino acids to give it its three-dimensional shape to still do the function, okay? And then also there are times when two amino acids behave the same even though they are different. So maybe the R groups here are behaving in this way because they're both hydrophobic R groups, so they go away from the water. But you might be able to substitute another hydrophobic, like if this is a hydrophobic amino acid right here with a hydrophobic R group, if you substitute it with another hydrophobic R group, you might still have that same interaction, and therefore it doesn't really change the shape. So that's where they're different amino acids, but they can still behave kind of the same and have the same effect on the overall structure and therefore function of the molecule. All right, so let's flip it over here. So this picture here shows you the wild type again, and this is, so the wild type, remember, is normal, what we see most often. All right, so here's your mRNA, and so we read it in groups of 3, remember. And this already did the work for you. Here's your AUG, that's your start codon. So, and remember, you always use those mRNAs in here. So they went A, U, G, and this is methionine or start. So methionine is the first amino acid. Then AAG, if you do AAG in the chart, AAG, you get lysine. So that's what I have here, and so on and so forth. So the order of the bases are supposed to, or order of the amino acids is supposed to be met, lys, PHE, and glycine. And then the last codon is stop. UAA is one of the stop codons. Do stop codons code for an amino acid? Absolutely not. So any of these three, there's no, it doesn't say stop or like it does met or start here. The stop codons don't code for an amino acid, so therefore glycine here is the last amino acid in the chain. So we're going to look at some examples of some base pair substitutions, substituting out one base for another. So there are 3 examples here, 1, 2, 3. So base pair substitution, there's no effect on the amino acid sequence. So when you look here, this is the normal here, and highlighted here is the U. So instead of GGC here, we have GGU. And so that's the mutation right here. And then if you look at GGU, all right, versus GGC in the chart, to do that here. So GGC or GGU both stand for glycine. It's the same amino acid. And so therefore, you still have glycine as that last amino acid, and then you have the stop codon. So this has no effect on the amino acid sequence. It's going to have no effect on the shape of the protein, and therefore the trait and the function of the protein. So this is a what kind of mutation? A silent mutation. Okay, because it did not, it changed the codon, it did not change the amino acid. Now let's look at what happens in a missense mutation. So again, it's a base substitution, so we're going to substitute one base for another. And so we're still looking at that same codon here. That codon is supposed to be GGC. And the G and the C stay the same, but they did an A instead of a G. So now the codon is AGC. So now if you look at AGC on the chart, I'm going back, AGC is serine. And so that's why I have serine here. And it's supposed to be glycine. So now we've changed the amino acids. Missense codes for the wrong amino acid. So the mutation when it happens codes for the wrong amino acid. So you have an amino acid there, it's just the wrong amino acid. Instead of glycine, it's serine. And then lastly, we have nonsense. And so instead of going with that same codon here, that codon's fine. We're looking at the second codon here. So notice here that the second codon is supposed to be AAG and stand for lysine, and the A is mutated to a U. So now you have UAG. UAG, if you read on there, on the chart, is one of the stop codons. It's one of the 3 stop codons, and so that doesn't even code for an amino acid. So therefore, at the second codon, you never even get to build the rest of your polypeptide. So now this is a really big deal because you've got a really screwed up protein in this case here, and so whatever it's supposed to do in the cell or in the body is not going to happen. All right, so you get a big side effect here. So here, the mutation causes a stop codon. Okay, so that's what we wrote here. Missense mutations code for an amino acid, usually the wrong amino acid. When it says usually, though, it could, like you could swap the third base. When it says usually the wrong amino acid, it usually always is the wrong amino acid. But you, like I said, if you could swap out like up here, and then it wouldn't be a missense, it would be one of those silent mutations. In a nonsense mutation, the wrong amino acid is encoded, and it usually results in a stop codon and the production of a non-functional protein. So, because you're not going to have a fully built protein. So these are often referred to, we're not on insertions and deletions yet. So these are your 3 base pair substitution types of mutations and the things that it can cause to happen. So now we're going to look at the second category. If we go back here, the second category, we had base pair substitutions, and now we have insertions or deletions. So this is not where we're substituting. So base pair, now we have insertions and deletions. We're not substituting, we're inserting an extra base, or we're taking out and missing a base. So these are often referred to as what we call frame shift mutations, because they alter the reading frame of the mRNA, and I'll show you that in a picture here in just a second. They almost always result in a non-functional protein. These are really bad types of mutations, and so you usually get a good side effect from that. So not a good meaning big side effect from it. Not good meaning that's not a good thing. The wild type. So let's look at here. So we're going to look at the same thing. Here's your wild type, and then we're going to look at the base pairs, insertion and deletion. And again, 3 examples, just like there were 3 for the base pair substitutions. So now we're looking at insertion and deletion. This is the normal. All right, so that we can compare it to. So let's look at here. What has happened here? So in this case here, they put in an extra U. The second, let's divide these up into codons here, every 3. All right, and so that eventually you get to the stop. The second codon is supposed to be AAG. So notice here, they didn't take anything out. Here's that second codon, AAG. Here's the third codon, UUU, and so on. They just inserted an extra U. So this is an insertion. So now what does that do? It shifts everything over to the right. So now the first codon is the same, AUG, but now the second codon is different, is UAA. The third codon is different, GUU. It's supposed to be UUU. So everything's shifted over once, so it affects every single codon after that. This particular one, it doesn't matter about that because look at what happens right there at the second codon. We have an extra U, and it makes that second codon one of the 3 stop codons, and so you never even get to finish your protein. What do we call not being able to finish it when the stop codon was reached? When we did over here, we call it nonsense. So this is a frame shift. Why? Because we're inserting an extra base and everything shifts. The frames shift to the right and change every single codon, but it's causing immediate nonsense, which means that the extra one immediately creates a stop codon and you stop making your protein. Now let's look at number 2, frame shift causing extensive missense. So now we're missing a U. So again, if we look at the third codon 1, 2, third is supposed to be UUU. So they took out that third U. So this is a deletion. The U is deleted, which means everything then shifts to the left. So the GGC shifts over, this G shifts over to where this U was, and everything shifts over. So now your third codon, instead of UUU, is UGG, and if you look at the chart, UUG stands for leucine, and then now the fourth codon is supposed to be GGC, now it's GCU, and so on. So everything after that deletion, every single codon is changed, and so you have extensive missense. What was missense over here? We coding for the wrong amino acid. And so that's what we have here. Once that deletion occurs, everything after every amino acid after that is changed because every codon is affected. And so you have a slew of the wrong amino acids there. And then lastly, the third type of insertion or deletion, it says insertion or deletion of 3 nucleotides. So an entire codon is taken out. So there's no frame shift. All the codons stay the same, but you're either going to have an extra or missing amino acid depending upon if you take a codon out or you add a new codon in. So first codon is AUG. Second codon is supposed to be AAG. If you take out that whole codon, now your second codon becomes what was supposed to be your third codon. So your second codon is UUU. Then your third codon is what was supposed to be your fourth codon, GGC. So that's why it says that there's no frame shift, meaning that the codons are still intact. They're the same codons, but they're not in the right order. So you're supposed to have methionine and then lysine, then phenylalanine. Now you have methionine and phenylalanine. And so therefore, in this case here, we are missing an amino acid. And that's why up here it says you could have extra or missing amino acids. So how do you get an extra one? by inserting. This is the deletion. This was a deletion up here, taking out a whole codon. But we could insert a whole codon, and then we'd have an extra amino acid. So all three of these are examples of what can happen with an insertion or a deletion. All right, and so those are your other types of mutations. So that's where I want to stop. This is the end of chapter 17.